Done. We're fine. Maybe I should also connect to power.
Do I need the microphone?
Hopefully, maybe I'll just stay close to you so that.
Or you can give it to me.
Okay.
Maybe, okay, okay, I'll just keep it in my hand. It's good now? Okay. No, it makes a huge difference. Distance. Okay. You're all set?
Yeah.
Okay. Okay, let's get started, everyone. Welcome to our 5th lecture in the seminar. Today, we have another recent Safari PhD graduate, John Firtina, who's going to talk about accelerating genome analysis. John is my PhD student who recently defended. When was it? In November. Yeah. So he's an expert in genome analysis and different methods of accelerating it in software and hardware. So feel free to ask him a lot of questions on this topic. This is a fascinating area with a lot of development, especially recently, on both hardware and software co-design as well as algorithms. And I believe there is going to be more in this application domain going into the future. Maybe it'll explode 10 years down the road. Who knows? Okay, John, go ahead.
Thanks a lot for the nice introduction, Onur. Let me try to attach this also to my shirt. Okay. Yes. Okay. I guess everything is set now. Cool. Yeah, as Onur said, today we're going to cover accelerating genome analysis. And if you are thinking what genome analysis is mainly about sending your sample to 23andMe and like getting some maybe information about your essentially your parents and so on. So this is basically more than that. So get ready to be surprised about what we can do with genome analysis. But before that, although Onur mentioned that I was a PhD student in Safari, currently I'm a senior researcher as well in the Safari Research Group and a lecturer at ETH Zurich. My research interests lie in the intersection of bioinformatics and computer architecture, and you can get to know us and our group using these links. So these slides are already available, as you know. So agenda for today, we're gonna first quickly cover what genome analysis is. So you're gonna have the basics of what a genome is, and then I'm going to be showing you the steps to analyze the genome, and then we'll move on to essentially our efforts to accelerate the genome analysis by designing algorithms and also co-designing algorithms and architectures. And then we'll quickly touch on some future opportunities for essentially newer technologies. So analyzing genomes is very important for many reasons. For example, we can uncover and also treat certain diseases by looking at your genome. Because, for example, we can even alter the content of your genome with genome editing. For example, if you have a mutation, if you have a certain code in your DNA that makes you prone to having a certain type of cancer, maybe we can target that region and alter it with genome editing to treat certain diseases. Or we can take a sample from here and detect harmful pathogens growing among us, and by doing so, we can also enable rapid surveillance of disease outbreaks, like very recently happened with COVID outbreak. There are also, of course, like many other different applications of genome analysis. So then what is a genome that we're talking about? Right? So every living organism and every one of us has a genome, which makes us look different and also have certain characteristics different than each other. So if you look closely to a genome, what you're going to see is this famous double helix structure of DNA. If you look more closely, you're going to see the pairs of molecules. We know that, or base pairs we know that guanine here, G pairs with cytosine and adenine pairs with thymine. And this DNA can be packed in a very compact form, making up the chromosome structure. In a single human chromosome, we can have at least 50 million characters or 50 million bases up to 250 million characters. And in a cell, in a single human cell, we have several of these chromosomes, and in total, around 6 billion characters in a single human genome. So this makes up our entire human genome. And we have trillions of these cells in our body. So in fact, we're essentially storing large volumes of data in our bodies when you consider these bases as characters. So then what we really want to do is we want to look at this full picture of our genome and try to understand what it really looks like, because even a single character change, even a single typo, almost in any arbitrary position in your genome, can rewrite your entire health story. For example, by a single character change, you can have an increased cancer risk, or maybe a sickle cell disease, or color blindness, or maybe somewhere in your genome we have a source code for improving the quality of your life. So the goal of genome analysis is really looking at this genome and generating critical insights from it so that we can solve, for example, fundamental challenges of life. So then when you consider the genome as a series of characters, this is essentially what you're going to see is a bunch of DNA characters coming together, right? We know that we have 4 characters or 4 letters in the DNA alphabet. And then when you think of how large an entire genome can be, I find this illustration is usually useful. So what this building is basically an Andreasturm. This is in Oerlikon, and we're located in the 16th floor of it. And if you look closely here, maybe you're going to see Nisa working over there, or someone else from our group. But then what I really want you to consider is like how tall this building is. So this building is around 100 meters long, and it has a few, several floors. And then if you want to essentially write every single character in your genome, character by character, to every line, in essentially a piece of sheet, and then when we stop, basically, when we finish writing your entire genome, what we're going to end up having is basically a bunch of pieces of papers, right? And if you want to stack up all these papers together, what you're going to end up getting is basically a pile of papers that are as tall as this particular building here, which is 100 meters long. And if you don't believe me, I did the math over there, which is written in very small letters over here. So you can check my math and you can see that this is correct, actually. And this is only for a single cell. So we have trillions of these cells. So you can think of how large or volume of data that we're dealing with. So then, since this is a very challenging problem to solve, yeah. Yes, we have 2 different characters, so this is a 2-bit, you can assume, in a DNA. What do you mean by, so in 2 strands? Yes. So if you have A in 1 strand, 1 line, you can have T. So what do you mean by 2 combinations exactly? To the next strand. But right now we're considering a single strand because remember I mentioned we have double helix structure of DNA. So you consider only 1 line of that DNA and then consider the characters like in a sequential manner, like written next to each other, without even considering what comes in the other strand, which is the complementary version of that character. So we can infer actually what comes in the other strand by looking at the other complementary strand. Are there any other questions? Okay. So we were talking about like how large this data is, right? And that also makes it very challenging to solve or reconstruct our entire genome from scratch, the human genome. And this was in fact a very challenging problem around 1990. So it took around 30 years for scientists to fully reconstruct the human genome from scratch. And people spent around $3 billion to generate that data and also to reconstruct this particular human genome. But even then, this human genome that we constructed within 13 years by spending $3 billion was not essentially completely full. So we had essentially certain gaps in the genome that we didn't know what it is. And this is mainly because of the issues in the technology and essentially the lack of the analysis tools that we had back then. So as the technology improved and as we also improved our algorithms and our understanding of the genome, people tried reconstructing the human genome from scratch again in 2018. And it took around 4 years for us again to fully reconstruct the human genome again from scratch. So this time, the estimation is we spent around few US million dollars. So you can then assume that it costed much less than what it costed previously, and it took much quicker to reconstruct the human genome, which was around 4 years. So then maybe you can already see a trend here. So the time is getting decreasing, and also the amount of resources or the cost is decreasing, right? And today, we can actually fully construct certain chromosomes from scratch by also using much less resources, meaning much less volume of data by spending also only a few thousand US dollars. So going into the future, the trend shows us that we're probably going to be able to reconstruct any human genome by using much fewer resources, maybe just spending a few $1,000s, and also the resources in terms of essentially in terms of the computational resources. Maybe it will only take a few hours rather than taking maybe 4 years or 10 years or whatever. So then there is this trend that essentially goes into the way of like making the genome analysis much quicker and also much cheaper. So then we were talking about the human genome a lot. So then what about the other species, right? So here what you're seeing is a viral genome. The viruses usually have much fewer bases. In fact, this particular virus has around 5,000 bases in its genome. And when you consider COVID genome, you have around 30,000 bases in COVID. So next, we have a bacteria over here. This is a particularly dangerous bacteria. You don't really want to get it, especially if your food is somehow like poisonous. And if you get it, you're going to have a very bad night. And hopefully, you're going to be limited by that, right? So the next morning, you're going to hopefully feel much better. But who knows? But this is a very dangerous bacteria. But this is a very small one. Still like has 5 million bases in it. And now we have human genomes have around 3 billion characters. So now, like, I also showed some trend in the previous slide, right? So the trend was analyzing the genome was getting quicker, and so it was getting cheaper. And maybe you're already seeing another trend here. Maybe as the genome size increases, maybe let's say the intelligence also is increasing, right? Who thinks basically this is happening that way? As the genome size is increasing, maybe the complexity of life is increasing. Or maybe if I'm asking this question, maybe the answer is already no, and then you're already prefetching that, which is actually in fact true, because what we have next is the onion, hopefully, which is much less intelligent than the human itself, and maybe, but we don't know. Maybe there are other like parameters to make the lives a bit more complicated. So onion has a few billion, 16 billion characters, and also plant genomes have much actually more characters. But really the takeaway here is that the genome size is essentially huge, and it's not even limited. We're not even limited by analyzing the human genomes. If you want to analyze plant genomes, the complexity or the volume of the data that you need to analyze is a lot larger than the human genomes. So then I also want to show this the famous essential central dogma of molecular biology. Probably you already remember from your high school. What we usually start with is DNA, which I consider as the source code of our genome. So we may have maybe some junk regions, maybe not so useful regions, although scientists are discussing today, maybe those regions are not so junk, but let's consider that they are not very useful in terms of the functions. So those, essentially, we also have some useful regions in the DNA, like we have useful regions in our source code. So those useful regions are usually transcribed to RNA. So we have certain enzymes that transcribe those regions. And then this is really like an assembly of the program code that you're writing, which then is executed in your processor to make essentially certain functions to make your application useful to to essentially generate certain outputs, which is what the proteins are doing after the translation step. So by analyzing also the genomes, we can identify whether you're prone to certain diseases. And how do we do it? So assume you have a control group where maybe you have without any diseases, right? So you know that these people don't show a particular symptom, but you also have another group of people where you call cases, where you know that these people are maybe prone to having a heart disease. And what you can do, you can essentially generate the genomic data of all these people and try to have some statistical analysis where you can try to maybe identify whether these people in the cases group tend to have certain mutations in their genome compared to the other control group where they don't have any certain mutations. And then maybe you can link those mutations to that particular disease, right? So this is a very simple statistical analysis that you're doing here. So this is essentially one example how you can do this analysis. And what you can also do, maybe you identify that you have a mutation that the other people do not have, and you're wondering what that mutation means, right? So you know the location of that mutation in your genome. And what you can do, we have these open databases, and you can essentially go to these databases and ask, oh, I have this mutation in this particular position in my genome. What does it mean? And it will essentially tell you some answers. So if you have, for example, that character instead of another character, then maybe you're prone to have hepatitis C or whatever. So you have essentially, you can use these public databases to understand about yourself a bit more. And of course, we have essentially other types of diseases that are linked to much larger variations. For example, you may be prone to having autism when you have a huge deletion of hundreds of thousands of characters in your genome. It's essentially a much larger deletion than having a single or few character change. So then, essentially, as I mentioned before, understanding and analyzing these genomes is really important, and the applications are basically only limited by our imagination. And I'll try to show you some examples. So one really fascinating example, in my opinion, is this genome editing. So potentially you're already aware of it. So the genome editing is a process where you can target a particular region in your genome and then alter the content of it. So this usually goes by either letting that region to have a random mutation instead of whatever the character that you have in your genome, hoping that you're going to have a different character than what you have. And then hopefully this will maybe disable the functionality that you have in your genome. Assume maybe you have, again, a mutation that makes you prone to having a certain cancer. Then you target that region, and then you sort of kill that mutation. And then maybe you have another character now which makes you less prone to having that cancer. So now we can do it in a programmable way. We can almost target any region in our genome. And people behind this idea who showed that this is programmable essentially recently won the Nobel Prize Award because it has many implications for treatment purposes. Also, very recently, we have these Nobel Prize Award, which was due to now we can essentially design or predict the structure of the proteins by using some AI solutions. And this is essentially very important because if you understand what a protein will look like, then you can also design drugs accordingly. You can design a drug such that it will attach to a particular region of the protein, such that it will make that protein maybe function differently, or essentially makes its functionality, it will disable its functionality, basically. So then this makes the drug design much more effective and much more quicker and so much cheaper by just simple the AI operations. And since this is also very impactful, people behind this also won the Nobel Prize Award again last year. So we also want to, of course, do large-scale analysis for thousands of hundreds of individuals in a quick way. People are also doing the genome analysis to have a rapid surveillance of disease outbreaks like COVID outbreak. Also, we're using genome analysis for testing purposes. People actually tried this with COVID recently. Instead of doing PCR testing, can we do sequencing-based testing? Or you can imagine maybe we can have drones where we attach sequencing devices to these drones, and we also maybe have some computation capabilities,
And then you can let these drones run in maybe in the agricultural areas, so that like they can monitor your agricultural area in a real-time manner, in a continuous manner, so that it can continuously maybe give you some insights about your agricultural area. Maybe it has some dangerous, it's growing some dangerous bacteria, so that you can have maybe quick actions on it. People are also doing genome analysis in space, but also we know that genome is a very valuable asset to protect. And I gave this example in the beginning of the lecture about this 23andMe. So if you don't know what 23andMe, this is a company where you would essentially, they would send you a kit, and you would essentially take a sample from your tissue. Maybe you take a swab, and then you send your own tissue to that company, so that they'll analyze your genome, and then it would give you some insights about your genome. Maybe it would tell you, oh, you're linked to this particular ancestry. Maybe you're prone to have this particular thing or not. But then what happened is that this a few years ago, actually, this is the third time probably I'm giving this. I'm showing the slide the third year in a row, or two years in a row, and I kept essentially saying that although we didn't know the impact, essentially what we assume, or what happened with this company, is that the data got leaked. So some hackers essentially were able to access this very sensitive data, and essentially they don't know what happened with that data. But now we actually know what happened with 23andMe. So 2 days ago, they actually announced that they are now broke. So people are not investing in that company anymore. And this is simply because this data is very valuable. And if you lose that data, then you lose the trust to the company. And now, and then you might be asking, so why is it so important that like maybe other people got my genomic data, right? So maybe you don't know the answer now, but maybe 10 years in a row, 10 years down in the road, you're trying to get an insurance, let's say. And your insurance quote maybe is 5 times larger than the average person. And you wonder why this is the case. And you don't know maybe why, but they know essentially why they are offering you a high quote essentially for your insurance, right? So this can be a huge problem, especially when you think of they can also attack a certain population by knowing their weaknesses. So then we really also want to protect this genomic data because it's very valuable. So can we also design algorithms or solutions that are in a privacy-preserving manner, because it's very useful. But then also we see these applications where you can shop with your DNA. And what does it mean? Maybe there is another company that we trust having our genomic data. Now what you can do, you can go to a grocery store, and then maybe you're trying to buy some bulgur, but you're not sure basically whether you're allergic to it. And what you can do, maybe you can just scan the barcode of whatever essentially the food that you're trying to find or buy, and then maybe this particular solution will tell you, oh, don't buy it because you're allergic to it. Or maybe, I don't know, it will tell you, oh, actually, you'll taste this and that or whatever, and then it will give you maybe some insights also linking with your genomic data. So essentially, you can now shop with your DNA, and this is a real company running now, which is still not broke, let's say. But you can also do computing with DNA. So you can design certain problems. For example, you can design DNA in a way that it will solve almost NP-hard problems. So people are now designing solutions to solve, for example, traveling salesman problem, because these DNA agents provide you a huge parallelism. You can assume you have billions of agents that can do the computation at the same time. So if you have such a parallelism, so it's almost like a non-finite automata, right? So you can assume like almost you have a unlimited parallelism over there. You can essentially go with almost any path in your graph and then try to solve very important, very difficult problems. And this is what people are trying to do with DNA computing. So these are only basically a few examples about what we can do with genomes and by analyzing also the DNA. Then I guess maybe it's time for me to slowly transition into to talk about the problems about analyzing this particular data. And one big problem is mainly because of this, essentially the general purpose machines that we're trying to use for data analysis. And this is because the data generators, and in this case, this particular machine that you're seeing here is a genomic data generator, and it is really specialized at generating data at a very fast rate. So it's really fast, so it can generate maybe terabytes of data at a very short time. But then we're trying to really use the general purpose machines to analyze this data where we don't know basically the requirements of the analysis of that particular data. And then we still need to analyze large volumes of data in a fast way. So the data moment is really a problem. So it is a problem because we know that a single memory request can consume more than 2 orders of magnitude energy than just doing a computation. So this is in terms of energy, but also takes a lot of time to move the data. So if you imagine you need to move the data across many stacks of the memory, many levels of memory hierarchy in the system, and then if you're moving essentially large volumes of data, it will take a lot of time until you can compute it. So then this means we really need to analyze, we really need to understand the needs, the requirements of this particular data, and then design algorithms, co-design algorithms and architectures that can handle this data well. And for this, we're essentially looking at different technologies, new architectures, and asking the question, given this particular data, so what sort of system should I have? Right? Maybe there's a particular application that makes it really suitable just to use maybe FPGAs or maybe just GPUs or maybe just a combination of all of these. Maybe we also need a heterogeneous system on chip. Or maybe we have a mobile device that also has certain requirements, such as limited battery, right? Then maybe you care more about the power consumption, the energy consumption, rather than how fast you're doing it, or maybe both. So to basically achieve all of these, we're usually looking at this entire computer architecture stack. I'm sure you've already seen this slide many times before. But this is really important, basically, to see the entire stack so that you can co-design everything all together. So if you're interested in essentially our efforts in our group, but also in the field in general, how people co-design algorithms and architectures to extract genome analysis, you can take a look at this paper. But we essentially have several directions for this genome analysis. For example, we're designing new algorithms to enable new directions in genome analysis. And this is one example where we try to analyze the genomic data in real time as it is generated from the data generators. So we don't even wait for the entire data to be generated. We try to analyze in runtime. So this is one example of it. So this is another example that also does it in real time. And we're not even stopping there. And what we're trying to do is we're also trying to reconstruct the individual genome from scratch, also by looking at the genomic data, and also try to find some mutations also in real time as we generate the data from the data generators. So this is another work where we try to deal with certain mutations in a fast and efficient way using some heuristic decisions. And in this paper, we also asked the question, what happens basically if the database that you're looking at is updated? So do you need to do all the computation from scratch? So I'm going to describe some of these in detail in the upcoming slides. So we're also utilizing some AI ML operations in genomics by designing algorithms and architectures together. So for example, this paper tries to identify the errors in the sequencing data, in the genomic data, by utilizing certain ML technique, by learning from basically the expected behavior of the data generator. And by learning from it, it can identify what sort of errors it will make. So this is an accelerator for the previous application, as well as also the protein analysis. I'll cover this also quickly. So we have also another work that tries to essentially translate the raw data that we're generating from the sequencing devices to text-based ACTGs. So for these, we usually use AI-based translators to translate the data. And also in this paper, we're trying to make this analysis much faster. And also for this translation, we also look at the processing in memory architecture and then ask the question, how can we do this analysis essentially using processing in memory? Maybe using some analog computations by also merging several steps in genome analysis. And this is another example that also uses analog computations to do the certain AI operations for genomics. We're also, of course, co-designing algorithms and architectures for the other steps of genome analysis. For example, this work accelerates the approximate string matching algorithm because this is at the core of genome analysis. I'll cover these again in the upcoming slides. This is another work that accelerates certain graph applications, again in genomics. So this is the first example of such a graph acceleration for genomics. We also essentially design solutions where we directly use storage, for example, SSDs, to do the analysis directly within the storage. So this is GenStore, like we're looking at such a solution in GenStore. And also, maybe can we do even more by combining the storage system with maybe CPUs and so on, and how can we partition several tasks for certain genomic applications by using both CPUs and also the SSDs. So again, many of these directions are covered in this particular paper. So if you're interested in, you can take a look at this paper. So now this was basically the overview of genome analysis and also all the efforts that we're doing in our group. So now I'll quickly go over some like step-by-step, like how we do genome analysis. So it usually starts by, the genome analysis stuff, it usually starts by taking sample from you. For example, we can take, I don't know, your blood or particular tissue from your body. And then we essentially prepare it for data generation. So what preparation means, so you fragment it, you sort of essentially break it into smaller pieces. This is mainly because of certain limitations in the sequencing devices that we're using, or again, certain biological limitations. And then you also attach some adapters to the end of these fragments so that the machines can read them. So this is known as library preparation. So you then input these fragments to these devices called sequencing devices. Then these devices will generate some data out of it. And these are essentially the computational steps that we're designing to analyze this data that the sequencing devices are generating. And to analyze this data, we have a bunch of steps. I'll go over those, but I'll start with the data generation. So what we're hoping to get is basically this one piece of like this maybe entire human genome in, again, one piece in a digital format, in a human readable, so that we can look at any position of that genome and then analyze it, maybe to generate some insights from it. Maybe you want to look at this particular position, and then what you're seeing here is a C instead of T, and then maybe you can link it to a certain disease. But then initially what we have is not something human readable, because we have a bunch of biological molecules. And we really want to basically go from here to here so that we can extract insights from it. And as I said, we can use these sequencing devices so that we can generate these raw data, and this raw data can be maybe electrical signals. And then we can use these AI-based translators or base calling techniques that translate this raw data into text-based ACTGs in a human-readable form. So now we have our human-readable data, but essentially I still have some bad news for you, which I sort of spoiled earlier, which is we don't essentially have this genome in one piece. So we don't have your entire genome or entire chromosome as a single piece. Rather, we have very small pieces that we call reads. And then this is, again, because of some biological and technical reasons. And these reads are extremely small compared to the entire length of your genome. And even beyond, we don't know the origins of these reads. So we don't know where they are coming from. So then this is really like a giant jigsaw puzzle that we're trying to solve. And since this is puzzle solving, we have a reference picture or a reference genome we can look at. But this reference genome we're looking at is someone else's genome. It's not your genome, basically. It's a pre-constructed human reference genome that I showed you before. So then, even though we identify the origin of this particular read, where it is coming from, we still need to accommodate for changes because of the mutations that this particular individual may have, or maybe the errors that we may have in the data. And of course, to cover the entire region of the genome, we're generating large volumes of data also redundantly because we have issues in the data. So that we can essentially reconstruct the individual's genomic picture, the entire genome from scratch by essentially mapping these reads to human genome and then seeing essentially what your genome looks like. So then when you think of it, this is really a huge data science problem that we're trying to solve that is usually in the volumes of terabytes or petabytes. And we have many different sequencing technologies, and they all have like different types of limitations, and they all essentially generate different types of raw data. For example, if you are using Illumina, it will generate sets of images that correspond to, depending on their color, it corresponds to whether you have A or T or G or C. And then by looking at essentially the intensity of the color, you can try to infer what sort of character that you have at a particular position in the read that we're generating. Or we can generate electrical signals that correspond to, again, the particular basis, which actually I'll cover quickly now what these electrical signals look like. So we're particularly interested in this technology that generates electrical signals, which is based on nanopore sequencing because it has many opportunities. And we're covering some of these opportunities in this paper, so you can take a look at this one. But really what it looks like, there's a very small device. So it's a handheld device. You can fit it in the palm of your hand. And what you can do, you can insert the DNA sample here, and then it will have essentially a bunch of these tiny pores called nanopores where the DNA will go in, and then eventually it will generate some electrical signals from it. So this is called nanopore sequencing process. It looks really nice, maybe like also maybe highly parallel because you have a bunch of pores here. But then it really didn't start like this. So it started with an idea. It started with an idea back in 1980s. Essentially, David Deamer was thinking about, so DNA itself is basically ionized. So it has certain ions attached to it. And then I believe he was thinking, can we create an environment such that we look at these ions and then try to tell what sort of character that we have at each position in this DNA. So this was the idea, and then the environment that he was imagining is basically this tiny region, tiny channel, where you try to essentially insert your DNA and then thread it basically through this channel, such that as this DNA moves, maybe the current that is also flowing here will be impacted because the DNA is also ionized. And by looking at this disruption or how this current is impacted, maybe you'll be able to tell what sort of phase that is passing through at that particular moment. So this was basically the idea, but of course, we had essentially many challenges. One challenge was how fast is DNA moving, and then can you control the speed of this DNA as it moves through this channel. So this was actually a big a challenge, and it took around 20 years to solve, around 20 years. But finally, we have essentially these devices that are really sophisticated, where we can control the speed relatively nicely so that we can essentially thread the DNA inside these channels so that they can move from the negatively charged side towards the positively charged side. And as they move, they will essentially disrupt the current flow that is flowing inside this channel. And then we will report these current disruptions as electrical signals. And the idea is that by looking at these electrical signals, maybe we'll be able to tell what sort of base that was moving at that particular moment. So what we can even do, we can start even analyzing this data as we're generating, which we call as real-time analysis. And even further, what we can do during sequencing, if we can understand whatever the DNA that we're sequencing at a time is not useful for us. Maybe you're interested in the human genome, but somehow the sample that you have is contaminated by maybe bacteria, and then you realize that that genome is coming from bacteria instead of genome. You can tell the sequencer, oh, I don't want to keep sequencing this read, so could you please basically stop sequencing it, maybe eject it. So this is essentially very useful because what's enabling is, it's enabling you to utilize these consumables for useful stuff instead of doing the sequencing also for something unnecessary, which you have to also pay the money and also the time for. So this is, to make these real-time decisions, then you need to be doing real-time analysis. So then what we have after nanopore sequencing is basically some raw data, which is electrical signals, and you have two solutions usually. You can either use these translators to convert it to ACTGs, or you can directly analyze these electrical signals without translation. So this is essentially the question that we ask. Do we really need to translate the data to do the analysis? So this is mainly because we know that the base calling is useful because we can make acute translation from electrical signal to text-based ACTGs. But then to do this translation, we need to use costly AI models. And of course, it can be fine to use these AI models if you have the computational resources to pay. But then maybe if you are working with a drone or maybe some device with limited computational resources, maybe what you want to do is, like maybe you cannot afford basically using these power-hungry devices, and maybe you want to do much cheaper analysis, right? So we asked the question, how can we make this pipeline cheaper? And one idea was basically, can we simply remove basically this translation from this pipeline? And of course, here, what we need to deal with is we have now two different languages. One is extremely noisy electrical signal, and the other one is text-based ACTGs, where we have our reference genome. So how do you enable communication between them? So to show you basically how this can be enabled, I'll quickly show what sort of electrical signals that we're generating. So this is a bit more essentially close look at how this nanopore is generating the electrical signal. So assume we're trying to basically sequence this entire DNA molecule. But at a time, due to the length of this nanopore, we can only fit certain characters, a fixed amount of characters inside this nanopore. And if you basically notice, so reading from backwards, so this is CTG, CGT, and this is basically the first 6 characters of this particular entire DNA molecule. So we can only fit 6 characters here because of the length of this nanopore. But then what you really need to know is that we will be generating an electrical signal collectively, essentially due to the presence of this 6 character inside this nanopore. And the idea is, as this DNA moves, so what happens is that one character goes out, here in this case is C, and the another one goes in. So we have the next 6 character or next 6 mer or q-gram or n-gram, whatever the terminology that you want to use. So given the 6 characters that we have here, now we have a different row signal. So we move to the next one, next one, next one, and so on. So as this DNA moves, we're going to have a series of signals. So what you can do, you can look at these series of signals, and then you can try to decode the entire DNA molecule that we were trying to sequence. But what I want to do is I want to sequence this DNA from scratch again, they're exactly the same DNA. So we start with the first one again. We move to the next one, next one, next one, and next one, and so on. So probably you already observed something here. So even though we sequence exactly the same DNA molecule, we did not generate exactly the same series of signals. So this is mainly because of certain types of noise that we have in the environment. But essentially, we also have a silver lining here, which is when we're sequencing the same DNA molecule, we're going to be generating similar raw signals. So then essentially the challenge becomes, can we identify basically similarity between these signals such that we can say, oh, these signals were coming from the same DNA, which is quite useful basically if you want to basically identify the origin of this series of raw signals in a reference genome. And one idea to do so, what you can do, you can represent these series of raw signals as n-dimensional vector, right? And you can maybe do some nearest neighbor search between them so that you identify maybe some cosine similarity between those. But then we identify this becomes too costly and does not really scale well, especially for larger genomes. And to solve this problem, essentially in RowHash, also in RowHash 2, what we did is instead of doing this costly distance calculation between signals, what we did, we quickly generated hash values from these signals such that we can match these hash values quickly rather than calculating the distance between them. But of course, the challenge is how do you generate the same hash value from similar enough signals? Right, so this is one challenge that we try to solve, and also we try to find only a few hash matches so that you're not doing a lot of computation. So, of course, like I'm not going to describe the details of the mechanism, but by doing so, we can enable real-time analysis for larger genomes, such as the human genome. And also in the subsequent paper in RowAlign, what we do, we utilize these mechanisms in RowHash, and we also even align the signals to each other so that we can identify the mutations between signals without even doing any translation. And in RowSample, what we do, we essentially push a bit more beyond, and then we ask the question, what happens basically if you don't have a reference genome that you can use? So this can happen, right? Maybe you don't have a reference genome that is available for an organism, or maybe that's not ideal. So in that case, what you can do, you can try to find overlaps between reads, between DNA fragments, so that by looking at these overlaps, you can try to construct the genome from scratch. So this is known as assembly, and people are doing this like very extensively in the field because we can generate critical insights from it. But then you cannot really do this without doing any translation. And doing so directly with raw signals is challenging because of many reasons. Because the signal is noisy, especially when, you need to find similarities between two noisy signals. You need to identify many useful overlaps, such that you can construct useful paths from them so that you can follow these paths to construct the individual genome from scratch. So we address these challenges in RowSample, and we essentially, when we look at the results where we compare essentially performance compared to you do this analysis without doing any translation versus doing translation and doing the same analysis, right? And then what we see is that when we do the same task without doing any translation, we achieve significant speed up because translation is costly, right? And we can achieve up to 60x performance improvement to do the same task without doing any translation. And this is based on using the same hardware, which is CPU in this case. But then even when we look at, essentially, when we compare our CPU implementation with the GPU-based translator, we see that we can still achieve speed up compared to the GPU implementation. So this shows that if you also have a GPU implementation of this analysis without doing any translation, the speed ups can be even much higher. So we construct also assemblies from using this overlapping mechanism in our RowSample work. And we see that actually we can construct essentially much longer assemblies such that they are almost half the entire genome length. So these are basically huge, and you can use them instead of basically base calling each read individually. Maybe you could just base call these assembled pieces. So this is, we of course explain many other details in the RowSample paper. So we also have other AI-based works where we try to translate these signals in an efficient way to text-based ACTGs, or we use basically some analog computation using some memory-centric devices in a processing in memory fashion, so that we do the ML operations using memory-centric devices, so that we do translation from electrical signals to text-based ACTGs as well. And this is the other AI ML-based work to do the error correction. So maybe this is a good time to stop before I start describing the read mapping step. So maybe we can take a 10-minute break, and then we can come back with read mapping. By the way, you can stop me anytime if you have questions. So feel free to do so. I know I'm going a bit fast because of the amount of content, but don't be shy. Oh, okay. Was it problematic? Okay. Sure. Okay, I guess you can hear me. Okay, so now this is really almost a half of the entire slide because I have around 200 slides, so an entire lecture, sorry. So we're almost there. So now, like previously what we discussed is there is certain types of problems with analyzing genomic data, and we generate raw data from these data generators, and we can use maybe either AI-based translators to translate them to text-based ACTGs, or directly analyze the raw data without translating them. So in the read mapping, the second step, I'll show you how we find the origin of these pieces, these fragments in the reference genome, what sort of data structures or the heuristics that we use to make this analysis practical. So as I said, this is really a giant jigsaw puzzle that we're trying to solve. We have biological sequences. These are reads. And what we could do to analyze these sequences, we could try to find overlaps between these reads, between these pieces, so that we can make sense out of the picture by looking at the overlapping regions, right? So you can, instead of having these randomly ordered reads, maybe you have some ordered reads, and then you can see what it really looks like. Or maybe you can take a look at some template sequence, which is a reference genome. And then by looking at it, you can also try to identify where each read is coming from. And this is really essential because we want to identify the origin of each read, exact differences, so that we can perhaps link them to maybe diseases or maybe do some other analysis. So this is how DNA logically looks like and how it physically looks like. And when we have these reads, so these are small pieces, and this is actually what we're trying to do. This is a reference genome, and then we're trying to identify the origin of each piece so that we can see your genome by looking at a reference genome. But then you really have millions or sometimes billions of these reads that we're trying to map. So then how do we solve this puzzle, right? A naive way would be you take each piece and then you start from the very beginning of the reference genome, and then you perform an approximate string matching algorithm, or we call alignment, because we're trying to identify the exact differences between this read and the entire reference genome. And then you need to do this for all the reads that you're generating. But if you wanted to do this, then this would actually take years or even thousands of years to do so. So maybe you could not really get an answer for your health, for yourself, before maybe you die, unfortunately. So then to get these answers much quicker, because they are critical, this is a critical analysis to do so, we apply certain heuristics, right, so that this analysis is practical. And here the reference genome can be billions of characters. In the human reference genome case, we have 3 billion characters. And the read is usually around few hundred characters or few thousand characters. So what we do, we first construct a certain data structure. So this can be based on, for example, a hash table, where we store all fixed length subsequences or k-mers that we have in the reference genome as keys in the hash table, right? So you can maybe take this CAA if you're using 3-mers or k-mers, and then you can use it as a key here in the hash table, and as a value, you can essentially store the position where this particular subsequence appears in the reference genome. And as a value, actually, we store the list of positions where each subsequence appears in the reference genome. So these are known as k-mers, as I said, and these are the locations. So these are useful because what we can do, we can also extract the same length subsequences from a read, and then we can directly and quickly query this hash table to identify where whether each subsequence exists in the reference genome, and if so, where they exist, right? So now maybe you have some candidate regions in the entire reference genome space because you were able to quickly find these regions by simply looking at this hash table, which theoretically takes O1 time to do so. But then further, you can eliminate some of these candidates because this is a heuristic, right? We're trying to find the exact match of very short subsequences where you may have it also in some other regions where this read may not be originating from because of, again, the heuristic decision that we're making. But again, maybe you may have several of exact matches spanning a particular region. So this may be really your actual region, the actual origin that the read might be originating from. So then later what you can do, you can go to that region that you really believe that this read might be originating from, and then you can perform your approximate string match only on those candidate regions, not in the entire reference genome space. So then really the goal of this approach is to reduce the essentially the cost to search from the entire reference genome space into only fewer regions, so that this costly approximate string matching can be done much quickly. So in blend work, we also asked the question, what happens basically, these exact matches are not really exactly matching, right? So here in the hash match, you're trying to find an exact match, but it may so happen that maybe you have a single character change. So in such a case, you're not going to find those exact matches. But maybe because of that, you're going to miss maybe informative information that will enable you to tell, oh, actually, this is the region that this read is coming from, but you were not able to maybe find this k-mer because of a mismatch, which can occur due to mutations or the errors. So in the blend paper, we're actually generating hash values such that these hash values are the same for highly similar subsequences, so that you can directly use these hash values to quickly identify similarities between certain subsequences when they are highly similar to each other. This enables us to basically, rather than relying on exact matches, we're now relying on highly similar matches to find even more sensitive candidate regions. This is also known as a local sensitive hashing that some of you might already be aware of. But then we also look at which of the steps in read mapping is really time consuming. And what we see is, even though we have these heuristics, the alignment, the approximate string matching step, and also this filtering step that I showed you before, which is actually the chaining step, is really the time consuming steps in the entire read mapping. This is mainly because we're utilizing dynamic programming operations for a large volume of data. In fact, this takes around 90% of the entire execution time of read mapping. So we also have some approaches to accelerate that, but I'll cover those later. But the other question that we're asking, do we really then rely on a single reference genome? Because a single reference genome is usually constructed either by looking at a single individual or a collection of few individuals that may not be representative enough of every essentially living organism or the entire population of people, right? So then this is problematic, because when you're trying to look at a single genome, what can happen is that you're going to have variants that will not exactly match to the reference genome. And for some of these variants, you're still going to be able to identify their origin, because we have these heuristics and also the alignment step and so on, so that can tolerate these variations a bit. But then if you have huge variations that your heuristics are failing, then you're going to miss basically mapping these pieces. So you're going to miss important information if you're looking at a single reference genome. So this is known as a reference bias, because this reference genome cannot represent everyone basically equally well. So then what is the solution, right? Maybe again, the naive solution would be, then instead of having one reference genome, we can have maybe n mini reference genomes, right? That represents maybe n different population. Then maybe you can look at each different reference genome one by one, and then you do the same analysis again for each of them, right? But then this is not really a very practical solution, because this does not scale well. So if you have n many reference genomes, then you have n times slowdown in your computation. So the another thing that we know is there is a huge similarity between reference genomes. So because between the same species, especially for human genome, roughly 99% of the time, almost the entire region, the 99% of the entire region will be identical between species. So maybe only that 1% that will make the difference. So then how can we leverage basically this similarity? So the solution is actually to use a graph. So maybe you can have a graph where you can represent the regions that the individuals differ by using maybe different nodes or different basically states, such that they will represent these variations. And for other regions, maybe you're going to have basically a linear connection of these nodes that will represent the reference genome with regions without any diversity. So this is actually how it looks like. So assume the first 3 characters of a reference genome is same across species, but maybe the next character can be either T or G. Now, like instead of creating 2 complete separate copies of the 2 reference genome, what you can do is you can just create a single node that shows you that difference, and then you can connect, basically merge into the region that is also still basically same between reference genomes. So this is essentially how the graph-based reference genomes really look like. And this provides basically a very compact solution for analysis because it can leverage the similarity within the same species. So this is the read mapping step by using a single reference genome or graph genomes. So regardless, if you are using a graph genome, again, the challenge is what is basically the region in that graph genome that you think that the read might be coming from, and then extract that region and then apply your approximate string matching. And then you can essentially use the output from read mapping, which is the origin and also the exact differences, in several steps, in several downstream steps. For example, you can reconstruct the individual genome from scratch in the assembly step, or you can identify the mutations that the individual has compared to reference genome so that you can link those mutations maybe to diseases, or maybe you have a sample where you have different organisms in your sample, and then you're trying to identify what sort of organisms that you have where you don't basically know previously, right? So this is basically a metagenomic step, and then this is useful to identify whether you have certain maybe harmful organisms growing and so on. So I'm going to show like this metagenomic step and how do we analyze basically many organisms in the same environment. So this is the metagenomic sample that we don't know previously, right? So you may have a bacteria or a viral genome in your sample, but you don't know basically what sort of organisms that you have. So then logically, it's just a bunch of DNA molecules for you. So what we can do is we can extract, again, regardless where they are, what sort of organisms or what sort of species they are coming from, we can extract k-mers from them, right? And then what we can do is maybe we can have a large database that includes the reference genomes of all known species, let's say. If you want to identify really what sort of organism that you have at a very finer granularity, you can have this huge database, or maybe you're just interested about what sort of bacteria that you have. Then you can have a database that contains all known bacteria, basically. Right? So this is a huge database. And here the assumption is then instead of having a single reference genome, you have multiple reference genomes because you don't know which organisms that you're dealing with. And then what you can do, you can simply repeat the read mapping steps on a very large reference genome, again, by trying to identify whether these k-mers are coming from a particular reference genome or not. So this is, again, becomes almost like an exact matching problem followed by maybe alignment. Then you identify, oh, maybe for these 3 organisms, which is a bacteria, a virus, and a worm genome, I guess, here, then maybe you identify you have these 3 genomes, and then you identify their ratios. So what is the ratio between them, right? Maybe the 75 or 25% of time you have a COVID genome in your sample, and the 70% of time maybe you have a bacteria and so on. And this is all done by matching these k-mers that you don't know what sort of organisms that they are coming from, and matching them to a huge database that contains all the reference genomes. And then try to find which reference genomes that you're hitting. That will be a clue for you what sort of organisms to identify what sort of organisms you have in your sample. So this is the metagenomics step. This is an example of how you can use read mapping in downstream analysis. So with that, I'll now start covering essentially our ways to accelerate these steps in genome analysis. So we know basically there are significant barriers in genome analysis, and this is mainly because when you essentially link all these different steps in genome analysis, which starts with the data generation and then translation, then maybe some quality control, read mapping, and maybe variant calling and so on. So these steps can, of course, change depending on the application that you're doing. So when we look at these steps, so this is like a pipelined computation, right? But then the throughput that each step, the computation throughput that in each step, or the data generation throughput in the very first step, is really different across steps. So then there is especially one particular step here, depending on the type of genome analysis that you're doing, that will bottleneck the entire pipeline. So then the question becomes, what is that particular step, and why are you bottlenecked by it, depending on our application? So here, for example, if you are using maybe the nanopore sequencing, which is the second row here, maybe you're bottlenecked by both the translation step and also the variant calling step. Right? Maybe then you can focus on, oh, can we accelerate the base calling and also variant calling, so that you can maybe match the throughput in the other steps, so that you have a nice flow of data here. Again, most of these are due to caused due to the data movement in the entire system, as I explained before, so I'm not going to explain it again. But the really takeaway here is that usually data is performed far away from the data. And then we need to really orchestrate the architecture really well, so that we can design architectures that handle the data well. And I'll show you one example of it. But before that, I'll give you a spoiler. And then for this particular solution, we use processing in memory technology. So I guess we already covered these in previous lectures. But again, if you're interested, you can also take a look at these materials. So they really cover what we can do basically with processing in memory technologies. But again, I want to focus on this particular work to show you basically a way or a work where we utilize this processing memory technology to accelerate a particular step in genome analysis. In fact, we're accelerating two steps in one design in genome analysis, which is base calling and the read mapping steps. So for this, I'll explain you the GenPip work. So remember, in nanopore sequencing, we are generating these electrical signals, right, as the DNA is moving. And usually we want to translate this data. So we translate them chunk by chunk, and then we have these text-based ACTGs. So these electrical signals are stored, as well as whatever that we're translating corresponding to these electrical signals are also stored. Then you do some quality control, that you look at whether this translation was successful or not. And then maybe remove some low-quality data. And then you store again the high-quality data. And then you use this high-quality data to perform read mapping, where you try to identify whether the read is mapping or not mapping. So they may not map as well if maybe you have a huge variation that the read mapping cannot handle, or maybe the translation did not really go well, and then we could not really filter it really well. So then the mapping results are also stored in the system. So by looking at these, we usually identify two main limitations. So first observation is, of course, we have multiple steps in genome analysis. And as we move towards these multiple steps, there will be a large data movement between these steps. But then we'll waste a lot of computation and also energy as we're trying to move this data, because some of the data will become useless for us in the coming steps. So I'll try to show you an example of it. So these are basically some rough examples, or a sample, essentially, the volume of data that we're dealing with. So the raw signal can be in the volumes of maybe 4 terabyte here. When you base call, the resulting data will be around, 500 gigabytes. And then we filter some of these and then perform read mapping and so on. So here you can see that there are many steps. So here blue is computation and the orange is the storage. But then across these steps, we're moving large volume of data. Right? So this causes a lot of latency, but also the wasted energy consumption. But then assume this step is after base calling is basically the 100% of the data that we're starting with to do the analysis. After maybe filtering, you filter 20% of them. So you have 80% of the data remaining. And then maybe after read mapping, again, 10% of them is not mapped. So you just have maybe, again, 70% of the data compared to what you initially started with. So then we moved 30% of the data unnecessarily, because although we realized that they were not useful for us at the end, but still you had to move it even though it was useless for us in the later step. So you wasted computation here and also the energy to move this useless data. So how can we basically understand whether the data will be useless for us as quickly as possible, and also how do we minimize this data movement in the entire system? So again, a naive way would be maybe you can design PIM solutions or processing memory solutions for each step in genome analysis, right? In fact, we already have these solutions. So we have a PIM accelerator for base calling, also PIM accelerator for read mapping. So these solutions essentially minimize the data movement for each step. But then how about the data movement between steps, right? So they are basically oblivious to the data movement happening between these steps. So then we identify that there is no prior work that handles this data movement between these steps, also that aim to reduce this unnecessary computation. To have an understanding of what will be an unnecessary computation, then you need to get to this step, right? You need to basically get here and then see what sort of read will be mapped and what sort of read will not be mapped. Then how do you basically do all of these steps and still try to minimize the computation here by looking at the results here, right? So there is some sort of dependency here. So how do you basically orchestrate the information flowing in these steps really well so that you minimize the unnecessary computation and data movement? So this is basically what we try to tackle in GenPip. And what we did, we made some assumptions. We first assumed that we have PIM solutions separately for base calling and read mapping. And then we looked at basically its speed up that this sort of accelerator could achieve compared to the regular execution of these steps. And we estimate basically around 2.7x speed up by having separate accelerators between these two. But then we also assume, let's have these accelerators, but then also we assume that there's no data movement between these accelerators, right? So then you eliminate the data movement between the steps. And then we notice that if you don't have any data movement between these steps, we can further improve the speed up. But then also let's go one step beyond and assume there's no data movement overhead between steps, but also there is no useless computation, meaning assume we realized that 70% of the data was useless for us. Assume that you start with 70% of data in the beginning. Right? So this was the assumption that we made, and then you can achieve even much further speed up. So this is basically a motivational study, right? So we did not design anything yet, but we looked at basically the opportunities, potential gains that we could achieve if we had such a solution. And motivated by those results, we essentially decided, oh, then maybe we should try to tackle these challenges and then design such a system that can eliminate the data movement between steps and also reduce the useless computation. So this is essentially what GenPip is doing. So this is an in-memory acceleration system which tightly integrates the two steps in genome analysis. It has two key techniques. One, chunk-based analysis. I'll explain these in the coming slides. The second step is, or second module is early rejection step that essentially stops analysis as soon as it realizes that this data will be useless for us. And by integrating all of these, GenPip achieves significant speed ups and also the reductions in the energy consumption. So then I'll show you what CPU or this chunk-based pipeline is. So the goal of CP is to reduce the intermediate data between these steps as soon as it is generated. So in the regular case, what we're doing is we retrieve the electrical signal chunk by chunk. And usually what you do is you base call each chunk one by one. And then after you base call the entire raw signal, you can assemble these chunks all together, and then you perform your quality control and the read mapping, right? So this is the regular genome analysis that I showed you before. So with the chunk-based pipeline, what we do, as soon as we have the first chunk from the data, we directly essentially perform the base calling, followed by the quality control for the chunk that we just generated, as well as read mapping. So we essentially perform these analyses for the limited amount of data we just generated, right? And then maybe after read mapping, you sort of have an idea what this data looks like. And then we can do the same operations again after we receive the second chunk, third chunk, and the other chunks, and so on. So then this is really like an overlapping the computation, the different computations, rather than doing all these computations in a sequential manner. So this is the chunk-based pipeline, and then by doing so, you can minimize the latency, reduce the latency a lot compared to what you would be doing if you would be doing all these analyses in a sequential manner. But there's also one advantage of it. So this is the saved cycles that we can achieve. So this is our entire hardware module. So maybe another thing that I forgot to mention here, maybe the early rejection mechanism. So how do we integrate the early rejection mechanism here? So what you can do is, as soon as you generate this read mapping information, you can use this information to ask whether this read will map to a reference genome or not. So you can already tell, maybe after the second read mapping or the first read mapping, maybe there will be some confidence level that you can tell, oh, this read is not really going to map because I'm not seeing any matches here, or I'm not seeing any matches here. So it's just better for me to stop here without even doing the next of the base calling, because you're extremely sure that after this cycle, this read is not going to map to a reference genome. So what you can do, then you can save cycles, because you do not do basically the steps, the third and fourth base calling steps, because you already realize that this read is not going to be useful for you. And so this is basically how you're also saving cycles, by the early rejection mechanism, by utilizing the information that you're generating at every step. So then this is basically how we integrate all this mechanism, the chunk-based pipeline and the early rejection. So here, what we really do is that, so remember I showed you that there are PIM solutions for base calling and read mapping. So we directly essentially use them, these solutions, in our hardware module. So this is the prior essentially base calling module, and also the prior the in-memory computation module. But what we also do, we enable communication between them. So this base calling module essentially generates the data. We have quality control, and then we send the data, we orchestrate the data to the read mapping module, and then get some information. And then if you realize that this read is not going to be useful, we can send the signal to the base calling module to tell that, oh, please don't keep base calling, because we realize that this data is not going to be useful for us. And so this is basically how the early rejection is working. So this is basically the integration of fusing of these two steps together nicely with the decision mechanism. So then we look at some results in terms of performance and energy consumption, and we compare GenPip, our work, to the CPU baseline, where we perform read mapping and the base calling using CPUs, GPUs, and also optimistic PIM, where in the optimistic PIM we assume there is no data movement overhead between steps, but still it lacks the early rejection mechanism and also the chunk-based pipeline. So then when we look at these results, we see essentially huge speed up compared to, of course, CPUs and GPUs, but also compared to optimistic PIM. So this actually shows the early rejection and also chunk-based pipeline is really useful beyond eliminating the data movement between the steps. So we also look at some energy results, and also compared to CPUs and GPUs and PIM, we again see significant improvements in the reduction of energy. Again, especially for PIM, because early rejection is enabling us to do less computation, meaning you're requiring much less energy compared to any other solution. So this is basically GenPip, where we try to integrate two steps in genome analysis. In Swordfish, so we have also a memory-based analysis of only base calling. So we perform. So this is similar to this solution here. It's another in-memory base caller. But then in Swordfish, we also integrate the noise that we have in the technology that we're using, which is memory-stir devices. These are ReRAM-based technologies where you essentially input the voltage to a crossbar array where you also have ReRAMs, which essentially you can use as weights. So basically, when you integrate the voltage, it sort of applies some Kirchhoff's law. You can sort of mimic the behavior of matrix multiplication there. But then this analog computation is noisy because of the technological limitations and so on. And what people usually do is they train the base calling techniques using maybe CPUs, like where you have nice computations with much less noise. And then they essentially integrate the weights that they train using CPUs into these crossbar arrays. But then this is problematic because this type of solution does not really assume or does not really know anything about the non-idealities of the technologies that you're using, right? So there will be some noise. So there will be noise due to the computation of analog computation and so on. So whatever the weights that you're using, they are not going to be ideal for that particular technology. So in Swordfish, we also integrate those non-idealities, the noise issue that we have when we're using these memory-centric devices, and readjust the weights accordingly so that our trained model is much more suitable for the particular technology that we're using. So we also have another computation in memory solution for food profiling. So there's another work that you can take a look at. Also, we perform approximate sync matching using PIM technology. So you can take a look at these as well if you're interested in them. So then the other question is, can we also process the data closer where we store them? And what I mean is basically the SSDs here, the storage units. And in GenStore, we essentially look at this question. So this is mainly because, so remember the Genome analysis pipeline that I showed you earlier. We already know that we store large volume of data in the storage systems, right? So it can be your SSD, and then in the next steps in the stack, you have maybe your main memory, the caches, and finally your computation unit, which can be an accelerator or a CPU and so on. But then what usually happens is that you need to move basically this large volume of data up until your computation unit so that you can process it, right? So then there's a huge computation overhead because the alignment is costly, and there's also data moment overhead. But then if you maybe design certain heuristics or accelerators and so on, maybe you can then minimize the computation overhead, right? So as you accelerate the steps in genome analysis, the computation overhead will be minimized, such that the data moment overhead will even become much more problematic, right? In terms of, especially if you think of in terms of the ratios that you're spending for computation and data moment, as you accelerate these steps, this data moment will become much more problematic. So then we ask the question, so how do we also minimize this unnecessary data moment in the storage system such that, again, the entire latency is minimized? So our key idea was, can we identify certain data, in this case is genomic reads, such that we can filter them before sending them to the processing units, such that we only send the data that we need to send, and the rest just stays in the disk. And this can be because of maybe these reads will become useless, similar to what we have in GenPip, or maybe we know that they are going to be extremely useful, but it's very easy to identify that they are going to be useful because maybe you can just do some exact matching and then identify the entire read is exactly matching in the reference genome. So you don't really even have to perform these costly alignment steps and so on. So maybe you can just do some very simple computations inside the SSD, which doesn't really require sending the data to the computation units. And yes, you can also do computations inside SSD if you use it well. And this is basically what we do in GenStore. So we address certain challenges in doing computation in SSDs, and such that we can minimize the data that we need to move in the entire system. So by doing this, we can minimize both computation overhead and also data movement overhead, such that we can achieve significant speed up and also the energy reduction. So if you're interested in how we're doing this, I invite you to take a look at this paper. So again, there's another paper in a similar, let's say, line, where we also look at how can we also use storage systems to perform metagenomics analysis. So if you remember metagenomics, so you have a sample, you don't know what sort of organisms that you have, But then this means you need to look at a huge reference genome, right? So which contains several different reference genomes. So it can be in the sizes of terabytes or even petabytes. So when you have such a use case, then this means you're storing even a larger volume of data, which may prevent you from storing this data in main memory. So you may really be essentially limited by utilizing the storage system where you can just need to move certain amount of data to main memory, essentially analyze it, move it back, they move another chunk of data to main memory and so on. So this can be really basic costly to do so. So rather what we do is, can we design an entire system where we essentially have a cooperative in storage processing for metagenomics analysis, where we co-design hardware and software such that we have a storage system, which can be an SSD, but also we have a host system, which can be, I don't know, your CPU or accelerator, where you enable communication between them, and then you do the computation also both in the host system and also in the SSD. Right, so this is again similar to JSTOR, but then you need to orchestrate really the host system and the SSD this time really well, because there are many steps in metagenomics. So in the first step, remember, we are extracting these subsequences from the sample. And the second step, we have our database that we're storing somewhere, in this case is SSD. So then you try to basically identify exact matches between those subsequences that you extracted and also the database, so that you can do some computation that gives you the abundance estimation. So then what we do, we look at all of these steps. We essentially look at the size of the data that we're storing, what sort of computation that we need to do in all of these steps. And then we introduce a bunch of optimizations. So of course, again, I'm not going to describe you each of the optimizations here, but what I want you to notice here is that, so we don't necessarily basically use SSDs to do all of these steps that I showed you previously. So it can be very well be the case that some steps are more suitable to do it outside of the SSD, such that you can overlap the latency, basically, right? So maybe you can do the computation, and as you do the computation, maybe you can still need to send the data. Then as you send the data, you can also start doing another computation in SSD, such that the data movement that's happening here is somewhat hidden. Right? So you can basically enable communication, efficient computation between them. So also this requires us, so how do you also store this large database in the entire SSD in an efficient way, so that you also maximize the parallelism, right? So you have many different channels in SSD where you can essentially access the data in a parallel way. So this means that maybe you can design an accelerator for each channel. Maybe here accelerator can be an intersection, right? So you have a subsequence here. You have certain subsequences in this database that you're storing in the storage. The computation that you need to do here is a simple intersection. So then how do you extract the subsequences in the host system and send these subsequences to SSD and retrieve the chunk of data from the database and perform the intersection, basically between the subsequences of your sample and also the database in a very parallel manner, by also maximizing the internal bandwidth that you have in the SSD. So here, basically, of course, if you really need to understand a bit more and then see how we're doing all of these, again, you need to take a look at the paper. But here, what you really need to understand is, to co-design the hardware and software together, you really need to know the limitations and the capabilities of the hardware that you're dealing with. So you cannot really simply do everything in one system and expect basically to solve basically all of the problems that you're trying to solve. And in fact, maybe here, maybe we're limited by a certain bandwidth, and then you design your accelerators according to that bandwidth limitation. Or maybe we're limited by the DRAM size that you have in the SSD, and then you design your solution according to that, and so on. But again, when we design basically this end-to-end genome analysis, we see again a huge speed up compared to the software solutions where we're using both cost-optimized SSDs and also performance-optimized SSDs. using performance optimized and using cost optimized SSDs compared to performance optimized and accuracy optimized solutions. Sorry for the terminologies here, but I hope it was relatively easier to follow. But of course, we're also achieving significant energy reduction because you're doing most of the computation inside the disk rather than moving it across many levels of the hierarchy. Here, we also show that we can achieve same accuracy as the accuracy optimized baseline while still achieving much better performance and energy reduction. So again, if you want to learn more about it, you can take a look at this paper. Given the remaining 8 minutes, I'll try my best to cover the remaining works. So in the next work, so we have Genome. Genome is an approximate string matching accelerator. So this approximate string matching accelerator can be used for many applications, but also it's a core step in genome analysis because it's used in the alignment step. So this is the first approximate string matching accelerator for the genomics. And here, what we really have is basically we have two modules. One module performs the distance calculation, which identifies what sort of basic differences that you have between a read and the reference genome. And the second module, by looking at these distances, it basically identifies the exact differences. Right? So this one tells you essentially the distance. So for example, it will have 2 differences, 3 differences, and so on, but we'll have some intermediate data which you can use to decode what sort of exact differences that you have, like at what position you have maybe insertion, at another position maybe you have mismatches, and so on. So in the distance calculation part, we have a systolic array structure where you essentially use the output from one processing and then essentially you transfer it to the other processing element because there's some overlapping computation between these steps. But then rather what you also do, you also store the information that you're generating each step in some SRAMs because you're going to use these SRAMs, this information also in the traceback step where you identify the exact differences in the second module. So then you're looking at the output that you're generating in these steps in the approximate string matching so that you can decode what sort of differences that you have. So these are basically 2 different modules that we have in Genome, and of course, as it so happens, we have impressive speedups and also the energy reductions and so on. This is Damla also explaining this work. We also improve this work by some software optimizations, but also integrating in FPGAs and so on. But we also integrate this Genome solution for graph-based applications. Here, this hardware module looks almost like Genome, but we make certain changes to it where we have, again, accelerators to extract, to sample certain subsequences from the read. But also, in the graph, so you don't have a linear connection, right? So you may have some differences, or you may jump from one region to another one. So this means that instead of having a simple dynamic programming computation where you have a simple dependency, you may essentially need to look back, essentially further beyond in the DP computation because of these the connections in the graph. So we handle these various connections by some hop queue data structure and integrated in the analysis in the accelerator. So then this becomes a first solution to accelerate sequence to graph alignment, again building on Genome but also modifying it so that it's applicable to graph solutions. So then basically we already seen that there are many accelerators for many useful applications, right? So we already know that we have accelerators for AI, right, because they are useful also. But we also have accelerators for genomics, again, because they are very important, right? So there's also an accelerator for the combination of these, as I also covered previously, for example, with Swordfish or for GenPip. We also have a basically look at basically the ML-based graph solutions for genomics and how we can accelerate them because they are also extremely costly doing so. So you already know that the benefits of using graphs in genomics. And they're also useful for many applications. For example, you can identify what's essentially how you can represent a protein family using some probabilistic graph structures. And then you can try to align an individual protein to these graphs to identify which family that protein is coming from. And by doing so, you can understand maybe its functionality and so on. Or you can maybe design probabilistic graphs to perform error correction, or again, to perform alignment and so on. So to do this, there is a probabilistic graph structure that is known as profile hidden Markov models. So this is basically used to generate the ground truth when training AlphaFold initially. So people use the multiple sequence alignment using these hidden Markov model structures, and then they use basically the information that we're generating from here to generate ground truth data. And this is basically an expectation maximization algorithm. It includes a bunch of functions, but also it has certain data dependencies. And this is basically a dependency between them, which you could exploit if you had a specialized hardware design. But if you're trying to execute it in the CPUs, it will basically take a lot of time. Actually, depending on the application, it can take almost the entire execution time of the application. So you can see how costly it is to use this ML approach for genome graphs. So in essentially one of our work in APHMM, we provide certain hardware and software optimizations to accelerate these. Unfortunately, I don't have much time to cover these steps. But what we really do is that we look at basically the connections that we have, for example, in the DP calculation, and then we design, for example, certain memory component, and then place them right next to the logic units, which are responsible to perform the computation for the dynamic programming computation, which is at the core part of the functions that I showed you earlier. Also, we design certain software optimizations. For example, instead of having dense calculations, we have a sparse DP calculation. But instead of to have these sparse calculation, you need to do some sorting. But then if you want to implement sorting in hardware, it can be really costly. So you replace it with some cheap filtering technique and so on. And this is how the entire hardware looks like. As it so happens, we have, again, impressive speed ups and also energy improvements and so on. So in Airlift, also we ask the question, what happens if the reference genome is updated? Right? So do we need to do the computation from scratch? The answer is no, because even if the reference genome is updated, the large portion of the regions are unchanged. So then we leverage basically this similarity between old reference genome and new reference genome and do only the computation only on those regions that is updated. So this is a pure software solution for genome analysis. So we're almost at the end. So I believe there are many opportunities for new applications, especially I like to ask the question, can we do basically full downstream analysis, starting from the data generation, read mapping, maybe variant calling, without doing any translation, by just maybe simply using electrical signals. So this removes maybe completely the translation step. Or can we basically cooperate the raw electrical signal analysis and the base code analysis together so that we do a much more useful analysis? We know that the things are also happening in industry today. We know the biotech companies who design these data generators, they're also placing accelerators inside these generators. This is Dragon, this is an FPGA solution that can do the genome analysis starting from the very first step, which is read mapping, up until variant calling very quickly as soon as the data is generated. So if you buy one of these machines, they come with these hardware accelerators, because basically we know that they are quite useful. And actually, there is another paper that was out very recently, a few months ago. They claim that they can do the entire step within 30 minutes. So this is quite an improvement, basically, compared to what we could do previously. So we also know that the tech companies are looking at these important applications, not just genomics, but maybe some other important applications. And then they look at what sort of operations that they need to accelerate, so that they can accelerate a large variety of applications, right? And in this case, this H100 is including specialized instructions to perform the dynamic programming operations quickly. And then they are even showing the benefit of having these specialized instructions in the case of genome analysis here. Of course, we're designing specialized hardware for very important applications such as AI. There's a wafer scale engine that performs ML operations, the Cerebrus engine. So then the question becomes, can we utilize these accelerators where we also have, where we use these operations in genome analysis. We also know that there are now these real DRAM modules that integrate processing capabilities inside these modules. So if you buy, maybe you cannot buy them today, but we know that they are real. Actually, there's a startup. Maybe it's not a startup anymore, but it's a startup in France. It's UpMem. So they have these DRAM modules that also come with basically these DPU chip, which they call it now AI chips nowadays. But they are essentially data processing units where you can do the computation right next to basically the banks that you have in the DRAMs, so that you can minimize the data moment latency. So this also shows the PIM is basically the PIM solutions. You're going to be seeing more and more PIM solutions in real world, and we're also part of these a project where we try to basically accelerate certain steps in genome analysis by using processing memory technology. So it includes many partners from IBM, ETH, Bilkent, and also the other partners in France and also in other regions. So if you want to learn more about like how we can accelerate genome analysis, you can take a look at all these materials where we cover again similar topics, but also many other things that we could not cover today, or you can take a look at this paper. We also organize workshops specifically on this topic jointly with computational biology or bioinformatics, top bioinformatics conferences. So you can see the bioinformatics folks are also giving importance to these solutions. We're also teaching courses on this topic. So this is, we have a very recent, actually, spring 25, it should be a project and seminar course where we are assigning projects again on this topic to the students. They are learning basically how they can design algorithms and architectures to accelerate genome analysis. So to conclude, we covered many ideas today to accelerate genome analysis. We want to do even better, maybe even for protein sequencing. So the protein analysis is approaching. So can we think of solutions for protein analysis as well? So I believe many future opportunities exist with the newer technologies, both in terms of emerging computational technologies, also in terms of data generation technologies. So with that, I'll end today's lecture, and thanks a lot for your attention. I guess we don't have any questions. Okay, good.